This is Lab Report 10, Biochemical Test 4. This is VTPB 426 Group 2, Diego Castillo, and myself, Rita Cantu Mahurin. In the introduction, we highlighted the purpose of this lab, which was to identify and differentiate bacterial species based on specific enzymatic reactions that reflected their metabolic and pathogenic characteristics. In the overview, we see that the urea hydrolysis test specifically detects the urease enzyme. And then we see in the CAMP test that it detects the CAMP factor, which enhances hemolysis, specifically with S-agalactate, and then also SRS when it's near this specific bacteria. The coagulase test identified coagulase enzyme and it converted fibrinogen. And so the fibrin clot confirmed that it's an SRS pathogenicity that is present. As far as our materials, we see that the media we utilized were 4 urea agar tubes. We also used 1 blood agar plate, and then we used 2 rabbit plasma tubes. In the cultures, we did use E. coli, P. vulgaris, SRS, S. epidermis, Streptococcus agalactae, and then Streptococcus pyogenes. Our procedures varied based on the type of test we were using. So in the urea hydrolysis test, we did streak each urea agar tube with E. coli, P. vulgaris, and our 2 unknowns, our unknown number 1 and our unknown number 2. And then we did incubate them, and the positive test was indicated through the pink color, and so this meant that there was alkaline pH from ammonia production. In our CAMP test, we streaked the SRS down the center of the blood agar plate. We also streaked the S. pyogenes 3 lines above it, and then we did 3 lines of S. agalactae below the SRS streak. And it was important that we didn't let the streaks touch so that we could confirm the positive or negative results accurately, and so the positive result was S. agalactae. And then the coagulase test, we inoculated each culture into the 0.5 milliliter rabbit plasma. We incubated once again, and a positive test result was indicated with the visible clot formation.
These are the results for the urea test. E. coli is a negative, P. vulgaris is a negative, even though it should be a positive. Our unknown 112 is a negative, and our unknown 292 is a positive. The E. coli was a negative because it lacks urease enzyme, meaning that urea will stay intact and pH will stay acidic in the urea. The P. vulgaris should have been a positive, though it presented as a negative, possibly because of a lack of bacteria on the actual slant. It appears there's no growth there, so the bacteria could have died when transferring. But it should have been a positive because P. vulgaris does produce urease, and it would have broken down that urea into ammonia, raising that pH and turning the color pink. For our unknown 112, we do see growth, but no pink color, meaning it does not have the urease enzyme, so it wouldn't have urea hydrolysis, and it didn't turn pink, so our unknown 112 is a negative. For our unknown 292, it appears as a positive because there is a pink color, which happens with that urea hydrolysis, which turns to ammonia, raising the pH. The initial pH of the urea is around a 6.8, but after the positive test, it'll be around an 8.4 or higher, making it more alkaline. So there, because of the ammonia production. This is the coagulase test. Our S. epidermidis is a negative and our SRS is a positive. The S. epidermidis is not coagulated, so it's just a normal liquid. But for the SRS, it is coagulated and it's a whole blob that sticks together. The reason behind this is because the SRS contains coagulase, which converts fibrinogen to fibrin, and that means it'll produce that clot. And then for the S. epidermidis, it has no clot, so that means there is no coagulase enzyme in there, and it just remains normal. This relates to pathogenicity because coagulase contribute to pathogenicity by enabling bacteria to actually form fibrin clots, and it'll shield itself from immune defenses and aid in abscess formation. It's a really key virulence factor for SRS. These are the results for our CAMP test. The bottom is the S-agalactae, and the top one is the S-pyogenes, and the central streak is our SRS. The S-agalactae should have been a positive with an arrowhead hemolysis that should have been seen. The S-agalactate produces camp factor that enhances SRS hemolysin. The S-pyogens should have been a negative because there is no enhanced hemolysis, and S-pyogens does not produce camp factor. And then for the middle streak of the SRS, it's the central streak, so it's the source of the hemolysin, and it provides that partial hemolysin that interacts with camp factor. The purpose of this test is to observe enhanced hemolysis at the intersection of SRS and the test organism that we want to see. Um, we should have expected an S-galactate to show an arrowhead-shaped zone of enhanced hemolysis near the SRS, and then the S pyogens would not have shown that. Um, our results were kind of expected, other than the S pyogens, where there was hemolysis where there shouldn't have been. I'm not entirely sure why. It could have been a result of just the bacteria that we used getting mixed with something else. Maybe there was some crossover contamination. It is kind of difficult to see in the photo, but there could be some errors in the inoculation of these bacterias that could have resulted in our skewed results.
In conclusion, we summarized the results that we saw with each test. So the urea test, the P. vulgaris with positive was a pink medium, and then the E. coli was negative. With the CAMP test, the S. agalitae was positive, and we also saw hemolysis present. And then with the S. pyogens, we expected a negative result. With the coagulase test, SRS was positive, so we did see the clot formation, and in contrast, the S. epidermidis was negative, and so we didn't see that same clot formation. The key takeaways were that the enzymatic reactions are, in fact, accurate diagnostic tools for bacterial identification. The urease, camp factor, and coagulase activity indicate distinct metabolic or pathogenic traits, and understanding these biochemical tests aid clinical differentiation of these various pathogens. Thank you so much. This is once again Group 2, the Castillo and Rita Cantu.